Hi everyone. In this video, I'm going to be talking about chemistry in everyday life. Well, this chapter does have the reputation of being the most theoretical chapter of class 12 chemistry, but I think this is the most interesting chapter also. And from my side, I will try to practice some questions at the end of each video, which will kind of make this a little more interesting, right? So we'll start on that note. Well, chemistry is all around us. Okay, we don't even realize it, but it is. For example, the food that you eat, the clothes that you wear, the water that you drink, your body, even the soaps and detergents that you use, and much more than that. So basically, everything around you, you're surrounded by the things and the principles and the molecules and the atoms, and everything else is nothing but chemistry. Okay, taking an example, that if we have any symptom, for example, a headache, what we do is we generally go to a medical store or a druggist, okay, and we take certain paracetamols or certain drugs, and we are good to go. The question is, what do medicines do? How do medicines actually work? This is something which we will be trying to understand as a part of this video. Also, I think this is a small question which I think I need to clarify, that when we go to buy medicines, right, it's generally written a medicinal store or a chemist or a druggist, right? The question is, what is the difference between a medicine and a drug? And is there even a difference? Okay, so let's start here. Well, a drug is chemical compound which is having a lower molecular mass. Okay, but the next question is that what do drugs do? Okay, so the drug moieties interact with macromolecular targets in our body. Well, we'll understand what these macromolecular targets are in a while, but the drugs actually interact with these molecular targets and they produce a response, okay? A response in our body. Well, this response, when this response is therapeutic in nature, okay, then it is called a medicine. But at the same time, we should understand that drugs actually are not very good when taken in higher quantities. So when the quantities of these drugs in our body is increased, that is when they become poisons, okay? So this is the difference between a drug and a medicine. The quantity is the main difference between a drug and a medicine and the type of response that it produces, okay? So the drugs are classified on the basis of 4 different types of classifications. The first one is on the basis of pharmacological or the therapeutic effect that they produce. The second one is on the basis of drug action, which means the biochemical process in which the drug can be involved. The third one is on the basis of the chemical structure or the type of atoms which are involved in the making of the drug. And the last one is on the basis of molecular targets. Well, the molecular targets are various proteins, carbohydrates, nucleic acids and lipids on which the drug actually attack or interact with, and we will be talking about this in a while in a much more detail. But these are the 4 different classifications of the drug moieties which we discussed about. Now, as we know, we want to understand how do drugs work in our bodies, okay? So, well, in our bodies, we have different kind of macromolecules which are proteins, nucleic acids and carbohydrates and lipids, okay? The drugs basically interact with these macromolecular targets, and these are called macromolecular targets or drug targets in our bodies, okay? So to understand how drugs actually work, we have to understand how catalysts work, okay? So to understand that, we should know what catalysts are. So catalysts are molecules which take part in any chemical reaction and they just do one thing, which is they speed up the reaction, okay? And they are also very highly specific in the reaction which we are taking part in, okay? And they themselves are not consumed in the reaction. For example, we know a very, very famous reaction. This is the hydrogenation of ethene, in which we have a nickel catalyst, and the ethene molecule here is called a substrate, and the ethane molecule which is hence formed is called the product, and the nickel metal here is a catalyst. What the nickel metal is doing, we'll understand in the next slide. In this reaction of hydrogenation, we need the ethene molecules to interact with the hydrogen molecules, right? So the nickel layer is just acting as a meeting point between the hydrogen molecules and the ethene molecules, okay? And because we want these two to react together, the nickel molecules will just form temporary bonds with these two, and these temporary bonds can break, and now the hydrogens can react with the ethene molecule to basically hydrogenate them, So the nickel layer is providing a necessary meeting point for these two molecules along with the functional group or the hydrogen molecule or the functional species which needs to attack. In this way, it makes a reaction much more faster. So the two things which the catalyst actually does is, the first one, we hold the substrate for the chemical reaction by making some temporary bonds, and the second one is to provide the necessary functional group for the attack for the formation of the product. Well, this is exactly what the drug molecule is going to do or is going to attack in a way. Okay. So catalysts present in the human body are called enzymes. Okay. So if they are organic catalysts or produced by the human body, they are called the enzymes. And there are some special proteins which are extremely specific about the function that they are going to perform. So why they are extremely specific, we have to discuss in a while. So these enzymes or the biological catalysts have special sites called the active sites, and these active sites have a very specific shape. Now these substrate molecules interact with the enzymes at these specific active sites to produce what we call an enzyme substrate complex. This enzyme substrate complex further results into the formation of the product. This phenomena in which the enzyme and the substrate interact with each other at the specific active site with the specific shape of the active site and the substrate molecule is called the lock and key mechanism. Just like every lock has only one key, similarly every enzyme can only interact with one particular substrate, and that is why this reaction of enzyme substrate is very, very highly specific. Now, let's imagine whatever product is formed in this reaction is nothing but the inflammation and pain caused in your body. And when that happens, you employ a certain kind of drug to restrict the pain. Now, let's understand what the drug is going to do. Well, the drug has to somehow stop the formation of the product, or the inflammation needs to stop, right? So the drug which you take can do this in two ways. The first one is by competitive inhibition. In this phenomena, what happens is that the enzyme has the same active site, and now instead of just one substrate which will bind at the active site, we have another thing which is the drug, which is called the inhibitor, because this inhibitor or the drug molecule is going to compete with the substrate molecule, and instead of letting the substrate molecule bind to the active site, the drug molecule is going to bind to the active site, and hence the formation of the product, which is the pain or the inflammation caused, is going to be blocked, because now the substrate cannot form with the enzyme, but the drug actually combines with the enzyme, and hence the pain and the inflammation is hence stopped. The second way in which the drug can do this is by non-competitive inhibition. In this case, again we have the enzyme and the active site, but now we have another site which is different than the active site, and that is called the allosteric site. In this case, the substrate molecule is again not going to combine with the active site. Before that, the drug molecule is going to combine with the allosteric site. When this happens, the active site is going to change its shape, and now even though the substrate wants to, it cannot interact with the active site, because now the shape of the enzyme or the shape of the active site has changed. Since now here the drug molecule did not directly interact with the active site but to the allosteric site, this is called the non-competitive inhibition, and again in this case the pain and inflammation is not caused or it is removed because the enzyme substrate complex was not formed. Now let's talk about how the communication inside the cell takes place. This what we see on our screens is called an animal cell. A typical animal cell is surrounded with the help of a cell membrane or a plasma membrane. Well, this cell membrane or plasma membrane does not allow the passage of some harmful materials inside the cell, but does allow the passage of some not so harmful or useful ones. That is why it is called selectively permeable in nature. Now let's understand how are messages transferred inside the cell. Typical cell membrane is made up of proteins and lipids, and we have certain proteins called the receptor proteins which are embedded inside the cell membrane. Well, these receptor proteins are responsible for the communication of messages inside the cell. As you can see, just like enzymes, they also have their binding sites or the active sites. And The substrate molecule, which in this case is the chemical messenger, which are special chemicals which are required for the transfer of messages between neurons and muscles. So these chemical messengers interact with the active sites at the receptor proteins, and when they do that, the message is transferred inside the cell. So the message is transferred inside the cell without even the chemical messenger entering the cell. That's how specific the receptor proteins are. In cases, the message that is transferred inside the cell is to generate pain or inflammation in the body. In those cases, we employ certain kind of drugs to stop that. Well, the drugs in this case will do something similar to what they did in competitive inhibition. Now, instead of letting the chemical messenger bind to the active site, the drug actually binds to the active site, and the message by the chemical messenger, which was formation of inflammation and pain, is actually blocked. Such kind of drugs which block the chemical messenger and its messages is called antagonist. Whereas in some other cases, the body lacks some important chemical messengers, because of which some important messages are not transferred inside the cell. In this case, we again employ certain drugs which basically do nothing but mimic the real chemical messenger, and hence the same messages which should have been transferred by the chemical messenger are actually transferred by the drug molecules. These drug molecules which mimic the chemical messengers are called agonists. Well, now, since we have done everything about the lock and key mechanisms, the enzymes and the drugs, let's try to test our knowledge. Well, the first question is, enzymes are highly specific in their action due to, the first is specific shape of active sites, specific shape of allosteric site, specific size of active sites, and specific size of allosteric site. Well, if we have understood clearly what I have taught in this video, we know that enzymes are highly specific because of specific shape of the active sites. So the forces that are involved in the holding of drug to the active site of the enzyme is, well, if you've read the NCERT, it's clearly written that all of the above forces, the ionic bond, hydrogen bond, as well as dipole-dipole interaction, can be used for this interaction between the drug and the enzyme. The third one says the term target molecules or drug targets is used in medicinal chemistry means. The answer to this is first, key molecules involved in certain metabolic pathways that result in specific diseases, not catabolic and not anabolic pathways. Now, since we are done with the questions part and the understanding part for the first part of this chapter, which is chemistry in everyday life, you can head off to EduRev, practice the test and read the notes. Thank you so much for watching.